Our speaker today is Paul Burton. Thank you, Paul, for accepting our invite and coming to this event. A bit about Paul before we start. Paul is a third-year PhD student at Miller. He is supervised by Professor Joshua Benjion. He received his master's degree in applied mathematics from the INS Paris Scala, under the supervision of Stanley and Nicolas Renier, working on 3D embedded graph to better understand the organization of brain vessel networks. His research seeks to develop machine learning methods for predicting the response of cells to perturbation, chemicals, and biologics. He is especially interested in taking inspiration from the causal inference literature as well as cell biology. That's a bit about Paul. Paul, thank you again, and the floor is yours.
Thanks so much, Prudencio. And yes, first of all, I'd like to really thank Cass and Prudencio for inviting me and organizing these amazing talks. So today I will tell you a bit about the Recover project, so efficient exploration of the drug combination space via model-guided in vitro experiments. So we have a preprint available online that you can check. And before, I think first things first, it was like a big project with outside collaborators. And so first, I'd like to really thank the Mila team and my collaborators, so Jerry, Deepak, Maxime, and Joshua, who have been working on the project at Mila. And I would also like to thank our outside collaborators, so Relation, which is like a UK-based company, and especially Jake Taylor-King, who has been handling the project from the experimental side of things. And we have also been collaborating with the Scripps Research Institute in the US, and they are the ones who actually perform the experiments. So let's dive in. So basically, in this talk, in this project, we really focus on drug repurposing. So we are not trying to develop new drugs or new molecules here. We are really focusing on drugs that already exist, and so and maybe use them for a different purpose as the one for which they have been developed. So this is called drug repurposing. And actually, as you might know already, developing new drugs is a long and expensive process involving many phases. So drug repurposing is really like a promising way to allow for a fast response, for example, in the context of a pandemic. So actually, this project was kickstarted at the beginning of the COVID pandemic, even if the focus has shifted a bit and is not on COVID anymore. And while single drug repurposing are promising here, we really try to explore combinations of drugs, and this is a way to increase our space of possibilities and to really tailor a specific treatment to a specific disease. And we think that's like a promising way to move forward. However, when you try to combine drugs together, the space of possibilities explodes, right? So it grows like as an exponential function of the number of drugs that you allow yourself to combine. And just to give you a few orders of magnitude, if we were to consider all the drugs that are approved in the US, which is around 12,000 molecules, and let's say we have a big lab capacity, and we are able to test around 5,000 combinations in vitro per week, which is a lot. Basically, it would still take us around almost like 300 years to search the whole space of possibilities. So you see that it's completely intractable. And this is not what we want to do, of course, because this is really time and cost inefficient. So another aspect, another solution would be to really rely on existing biological knowledge, and that's what people have been doing. That is to say, work using the known targets of the drugs, understand the pathways that are involved, and really focus your attention on a small and restricted part of the space, in the part of the space that we understand well. But of course, we forget about lots of possibilities which may be promising as well. And here in this project, really, we want to use machine learning and more specifically sequential model optimization to try and balance between exploitation and exploration, as I explained. So we want to search promising solutions or drug combinations given what we already know. So biological knowledge, but also past observations. And we want also to explore widely through the space of possibilities. And basically, so sequential optimization, this is the terminology that we chose. You may have heard about active learning or Bayesian optimization. So here, sequential model optimization is a slightly more general concept. And basically, it tries to basically iteratively search a space of possibilities to find a maximizer of a given objective function. So in our case, that would be the synergy of a drug. And oh, I forgot to say it before, but do not hesitate to ask questions. I really like when it's interactive during the talk, so don't hesitate. All right, and so basically what's new in this projects is that we apply sequential model optimization to human cells. So in the context of drug combinations specifically, but to the best of our knowledge, that's the first application of these iterative methods to human cells. And it's quite difficult because human cells are less stable than other organisms, such as bacteria and yeast. And we had to deal with problems such as batch-to-batch variability. So that's for the introduction. Then let's say a word about the methods. So here, this is really the big picture about the project. And really the machine learning part per se is we kept things rather simple in the end because we had so many things to deal with that we really chose to keep the machine learning simple. And also it's a way to ease the adoption of the pipeline by other practitioners, which may not have experience with complicated graph neural networks, for example. So basically on the left-hand side, you can see the machine learning pipeline with dynamic training set, which is composed of so a set of drug combinations that have already been observed and their synergy. We'll explain what synergy is in more details in a second, but basically that's a scalar that is associated with a given drug combination, and so higher is better, and that tells you whether the two drugs, this combination is interesting basically. And so our model per se is like a modified version of an MLP with a few tricks that we'll explain in more details later. And so this model is used to, so it's trained on the data that has been acquired so far, and is trained to predict the synergy of new drug combinations, along with some uncertainty estimates. And after that, the predictions of the model and the uncertainty estimates are fed to an acquisition function, which is used to score the different combinations. And then all the possible candidate combinations are ranked according to this score. And the top 30 in our case are sent to the wet lab for evaluation. So the experiments by themselves take around like a few weeks. In our case, it took like something like a month to do each loop because it takes time to wake up the cells and find the right drugs, things like this. But basically, so the cells are cultivated in vitro and put in contact with the two drugs at various concentrations. And so for each queried combination, basically, there is a dose response matrix that is acquired, wherein each drug is tested at various concentrations. And for each pair of concentration, we measure the percentage of cells that have died because of the two drugs. And this is what we call viability. So in our case, we were focusing on cancer cells, and the goal was to, we were using primarily anti-cancer drugs with the idea to find combinations of drugs that are effective against cancer. And still, as a way to keep things simple, basically, we compute the synergy based on this matrix and summarize whether the combination is good or not with a single scalar. And the acquired batch is sent back to the machine learning pipeline and added to the training data set. And the whole process repeats again. So let me say a word about synergy in a bit more detail. So we've been working with a public database, which is called Almanac. And it involves a bit more like around 100 drugs, as you can see on the right-hand side. And basically, pretty much all the combinations among these drugs have been acquired. And we have access to a synergy score for all of them. And what is interesting to see is that the synergy has like a long tail towards the positive. That is to say, the region of interest is towards the very positive synergies, but that represents a really tiny portion of the whole data set. So that it was a bit tricky to deal with this aspect. And so synergy, so I mentioned the viability before, which is the percentage of cells that die. And basically, the synergy is the difference between the viability that you expect given the individual effects of the drugs minus the viability of the drug combination. And in that case, the viability that you expect is the product of the single drug viabilities, which is similar to independence in the probabilistic sense of the term. And another thing that we noticed is that there are huge batch effects between studies. So we also used the O'Neill study, which is a different study from the main study Almanac that we used. And basically on this plot, it's a scatter plot of all the combinations that are found in both studies. And you can see that it's a bit all over the place, right? So there is not a lot of agreement between studies. So there are a few differences in the protocols of both studies. But still, like you can see that there are huge patch effects. All right, let's say a bit more about our model, which again was rather simple. So it's mostly like a regular, fully connected neural network. And in some cases, in some experiments, we wanted to condition on a cell line because we had access to data coming from different cell lines. So a cell line being a specific type of cell, basically. And so we used film conditioning, which you can think of as a scaling and shifting of the activations inside the network based on a cell line representation. And depending on context, we used either a one-hot encoding of the cell lines or cell line features based on genetic variations and base level of gene expression of each cell line. Another aspect that was important to consider is the fact that we really want our predictions to be invariant per permutation of the two drugs. It is to say having drug 1 and drug 2 should be the same as having drug 2 and drug 1. And for this, we use the bilinear merge, which is just basically a bunch of dot products with like symmetric matrices. So a tensor, and each slice of the tensor is a symmetric matrix, just to ensure that the output of the network is invariant per permutation. And on top of that, as mentioned before, our pipeline needs to have access to uncertainty estimation methods in order to estimate the uncertainty about the predictions of the model. And for this, we were experimented with two different methods, deep ensemble, which is widely used. And so it corresponds to initializing several times the same network with different weight initializations. And then you consider the prediction of each specific network as a sample from the predictive distribution. So the predictive distribution is basically the distribution of synergy that you predict for a given combination of drugs. And another method that we used was to directly estimate the uncertainty using a second model. So in that case, we initialized two models, one mean predictor to estimate basically the expected synergy for each combination of drugs. And so this model was trained with mean square error, like in a very standard way. And we also initialized a second network, which was the uncertainty predictor, and which is trained to estimate the standard deviation of the predictive distribution. And so it was trained with this specific criterion, which is a way to get an estimate of the standard deviation of the predictive distribution. And actually, we did a bit more work on uncertainty estimation. And so you can also check this paper, Duke, so direct epistemic uncertainty prediction. And so it's very similar to what I just described, with a few additional tricks, because we focus on the epistemic uncertainty instead of the total uncertainty. And also we estimate the uncertainty out of sample. So we have a separate training set for the mean predictor and the uncertainty predictor. But basically in this work, we showed that directly estimating the uncertainty worked better than ensemble, and basically our estimates correlated better with the residuals of the model, and the order of magnitude matched the actual residuals of the model better. All right. And so once we have our model with our uncertainty estimates, we use the sequential model optimization, which is really simple, but let me go over it briefly. So we have like a data set of observations that have already been made. And so at each iteration, basically we have a set of candidate combinations, denoted X here, and an estimator that has been trained already. So our models in this case, and we have an acquisition function alpha that we use to score all the possible combinations, and we find the argmax of this acquisition function, or let's say the top k in our case. And the top k combinations are queried and sent to the lab for experimentation, and we query the synergies of these and add these observations to the data set, and the whole process repeats itself. And in practice, we experimented with several acquisition functions. So let's list a few of them here. So first, there is pure exploration, where yes, Prudencio?
I was wondering, when you add the acquired sample to the training set, how do you handle the distribution shift that's happening?
So basically, in our case, there was, so we made sure that there was not too much distribution shift, in the sense that the distribution of synergies is pretty much the same all the time, even though there are batch effects between studies. So we did not have to normalize the synergies, for example, or things like this. So there's a whole processing happening to make sure that the synergies are always within the same range. Does that answer your question?
Um, it ends up for the target part, but I think there's on the input side part,
Oh,
there's also a decision shift happening, right? Because then you explore different part of the big matrix that you have shown at the beginning, and yeah, just wondering how you deal with that.
yeah. So actually, in that case, really things are quite simple. And actually, when we perform the acquisition, we don't acquire the input features of the drugs. So the input features that we use for the drugs are the Morgan fingerprints. So it gives you, so it's a binary vector, and it gives you information about the structure of the drugs. And it's something that you can compute. Yourself. So you don't need to acquire it. So you don't have basically distribution shifts in that case. To be a bit more precise, the shifts that we are experiencing in that case is like a concept shift. That is to say, the distribution of the input X does not change. But the function that you are trying to estimate, so the function mapping X to synergy, changes from one batch to the other. So it's different from like a covariate shift, for example. And that's it was so the reason why it changes is a bit it's a bit unclear, but it's mostly due to the fact that the human cells are unstable and they may be in different states from one lab to the other, or maybe there have been a few mutations between the cell lines that are grown in different labs. But we really don't have to deal with covariate shifts or shifts in the distribution of the input features because they are all computed analytically, I would say.
Thank you. Good. There's one question by Vincent in the chat that you might want to address.
Sure. So why Vincent? Hi Vincent. Why is it always
C'était sur les... Sorry, it was on the like when you define the bliss values, you
yes.
had a difference between like the kind of uncorrelated expected value and possible synergy. But then, so you had negative, I mean, it could be possible that you had negative values. It was a while ago, sorry. Yeah, on the scale on the right, you only have positive values. I was curious as to why this happened.
Yes, so that's a good point. Thanks, Vincent. I actually forgot to explain this part. So basically, the bliss synergy is basically computed for each pair of concentrations, and basically we end up for a given dose response matrix, we end up with a matrix of bliss synergies, and then the question is, how do you aggregate all these bliss synergy scores together in order to obtain like a single scalar? So we experimented with either average pooling. So in that case, that would be the average bliss synergy across the matrix. In that case, it would indeed be centered in zero. In the end, we chose to focus on the maximum synergy score. That is to say, you use max pooling over your matrix, your dose response matrix. And so that's why it's always, almost always positive. I mean, it's like the mean is above zero because you keep the max over the matrix. Does that make sense?
Okay, but why do you test all concentrate? I mean, okay, that's another problem maybe. Like it feels like you take a max, but you could have taken the max of, I mean, like the max on each axis instead of the max of the matrix when you use them separately, because maybe you have like one response for one drug at a given concentration, one response, like a max for drug A, a max for drug B, and a different max for AB. And so here it looks like it, yeah, I mean, I don't know. So maybe
so you don't know where the max will be in advance, right? So you need somehow to acquire the whole landscape. So In practice, what would be ideal would be to basically feed this whole landscape of viability or synergies to the model. But as an attempt to keep things as simple as possible, we just decided to summarize it with just one scalar. And of course, there is a lot of information that you lose along the way. And we could come up with like fancier ways to do the acquisition so that we don't need to acquire the whole matrix. But still, for now, things were really simple. So we were doing the acquisition of the whole matrix and then just computing the max synergy. Then we could, for example, try to estimate where the maximum synergy would happen inside the matrix and just perform the acquisition of this submatrix. But that gets trickier, I guess.
Okay. Anyway, I think that answered my first question very clearly.
Okay, cool. Thanks, thanks for the question, Vincent. Any other question? Yes, Emmanuel? Or Hossein, sorry. Who wants to go first?
Okay, thank you. I'm Hossein. Hi. And just I have a question about your Bayesian optimization or sequential optimization scheme. What is the search space of this sequential optimization, the pseudocode that you showed? The X just contain the two drugs, or the X space also contain the two drugs and also cell line?
So it's a good point. So it depends on the experiments. So in some experiments, mostly the in silico ones, there was the cell line as well. That is to say, you choose a specific drug combination, so that you have the two drugs and so the cell line. So it's a triplet that you query. In the actual in vitro experiments that we performed, we were just experimenting with a specific cell line, MCF7. So in that case, the search space was just basically pairs of drugs.
Thank you. Thank you.
Welcome. Yes, Emmanuel.
Thanks, Paul. I have a quick question about the acquisition function. So if I understand correctly, you were taking the top K of a value returned by the acquisition. Okay. So have you looked into using more batch acquisition function? I know, for example, there's a batch bolt. I'm asking that because I'm kind of a bit worried about the diversity of your batch during the each iteration.
So we actually experimented with batch bolt. So to give a bit of context, batch bolt is like an acquisition function which tries to when you query batches of drug combinations, in our case, trying to make sure that the combinations that are queried are diverse and not all focusing on the same subpart of the space, for example. So we did experiment with this, and I think there was not much difference with Silico methods. So we, in the end, what I'm presenting here is like quite simple on the machine learning side, but we have tried many things, so fancier acquisition functions and also fancier models. In the end, we just kept everything simple because this was just what works best or as good as fancier things. Then I think these things are like super context specific. So in another context, even still drug discovery, batch bolt may outperform greedy or UCB by a large margin, but in our case, we didn't see any difference.
Thank you.
Thanks. All right, so I think I had not had time to explain all the acquisition functions, but so basically, so pure exploration is the case when your search is only driven by the uncertainty of the model. And so you're trying to query examples that are informative and that corresponds to the context of active learning. Then there is the other extreme where you follow pure exploitation strategy. And basically your acquisition function only cares about the expected synergy of each combination. Then there is like the random approach, that we call brute force, where you query everything at random. And then there is a UCB or upper confidence bound, which balances between exploration and exploitation and takes both the expected synergy and the uncertainty into account. All right, so I already said a word about this, but basically we tried to leverage protein interaction databases because if you think about it, we are trying to make predictions about the synergy, so the interactions that happen between the drugs and like also their downstream effects that happen inside the cell. So we need to capture and to understand something about the interactions that happen inside the cell. In practice, we could not manage to obtain a significant improvement in performance compared to the simple models that I presented earlier. And it may be due to various things. My main guess is that it's due to the fact that first, the task is quite simple. So it's a simple one-dimensional regression task. And also the diversity of the dataset was not that big in the sense that we only had access to 150 to 200 single drugs. So even if the space of combinations is used, the single drugs were not that many. But this needs further investigation. And if you're interested, I can tell you a bit more after the talk about the specific models that we explored. But let's, so yes, Denise.
How did you try to combine this protein information with in your model? Because you have this network. How did you try to use it in your model?
So we used the graph convolutional neural network on the graph of protein. So each node is a protein, and we had messages passing between proteins, and we also had some nodes that were drugs in our graph. And they had other types of messages, but basically it was like a multi-layer GCN. If you want, so I have a few slides about this if you're interested, but I'm not sure we'll have time to talk about this during the main talk. So if you want, we can catch up towards the end of the meeting.
Okay, thank you.
All right, so let's say a word about our Silico evaluation of the models. And so we took a lot of care regarding the evaluation of the model, and we really wanted to understand in which context basically the predictions of the model would actually be like predictive, and which context it wouldn't. And that was also a way we used this evaluation as a way to design the in vitro experiments that were performed in the lab. So we came up with 5 different tasks. So with corresponding to different ways to split the train, validation, and test sets. So there is the default one where basically you split the train, validation, and test randomly among a given number of drugs. Then you have the drug level split. So you test the ability of the model to generalize to combinations of new drugs that have not been seen during training or validation. We also tried to see the impact of taking into account several cell lines. So basically our models was trained on various cell lines and tested still on MCF7 only, which is the cell line of interest in our case. We also tried to see, we wanted to see if we could transfer to a completely new cell line altogether. And also, again, wanted to measure somehow the batch effect between studies. So training on one study and evaluating on another one, and still making sure that the test set was among drugs that had been seen in the original study. So overall, basically, we obtained a reasonable performance for the default task, and we even noticed that there was a small improvement in performance when taking into account multiple cell lines or generalizing to new cell lines. However, the performance completely dropped when trying to make predictions for new drugs or when transferring between studies. So you can see that we here the R squared was around 0.4 and drops to basically 0 on the test set, and more or less the same here. So the striking thing here that you might tell me is that, all right, all these metrics seem extremely low. And yes, it's true. And it's basically due to, we argue that this is because our observations are extremely noisy. And as a way to measure this effect, we basically tried to measure, to compute like an upper bound for the Spearman correlation and the R-squared as a function of the level of noise in the observations for a data set for a regression task where the distribution of the targets followed the same distribution as the synergies in Almanac. And we also evaluated the true level of noise in our observations because we had access to replicates of so several experiments had been performed for the same drug combination. And the red line is the performance of recover. So we are still not at the maximum, but you can see that a big part of the bad performance can be explained by the noise. And what is interesting is that our models actually captured very well the tails of the distribution of synergies, which were the regions of interest. And that's why in the sequential experiments we performed. Yes, Yves?
Yes, hi. So I was wondering if my experience is as well that these observations are quite noisy, but some data sets are better than others. So in particular, the O'Neill dataset, I think it has 4 replicates or 6 replicates for all the monotherapies, and it has 4 replicates for all the combination viabilities. But the Almanac is performed with quite few replicates, I think, for the combinations they're only done once. And then in particular, since you're taking the max synergy, you could imagine that the effect of outliers would be quite large.
Yes.
So I was wondering if you looked at anything in from your previous slides on the metrics, if you tested the model using only the O'Neill dataset, and if you saw some sort of quality difference in that.
So as far as I remember, so we did not perform this analysis on the O'Neill dataset. So this is only on the Almanac dataset. On the O'Neill dataset, we did try to train some models, and the performance were more or less the same as far as I remember, but it's not reported in the paper. But thanks. It would be interesting to explore this point. Yes.
Cool. Thanks.
Thanks so much. I'm looking at the time. Maybe I'll try to finish the talk before taking other questions, because there are still like important parts that I have not covered. If it's fine for you. So basically, so after this out of distribution capabilities analysis, we actually simulated the sequential model optimization experiments. So in Silico, and in that case, we saw that so what you're looking at here on the left-hand side is the cumulative proportion of the best combinations that have been discovered in our data set. So higher is better. And so the blue curve is a random search, and green and orange are 2 model-guided searches. And you can see that they outperform the random search by a large margin. And here we put ourselves in a context that matched the planned in vitro experiments as close as possible. So we were querying 30 combinations at a time, and the size of the search space was more or less the same as what we were planning in the in vitro experiments. And we used the simple models with deep ensemble as uncertainty estimation. And on the right-hand side, you can see the average synergies of the combinations that have been queried, and you can see that even pretty early on, you can get an enrichment. So that was quite promising. And however, you can see that in these experiments, we did not see any difference between UCB and greedy. However, in some cases, in some other experiments, especially so you're using direct uncertainty estimation and also having queries that were smaller, so querying 5 combinations at a time instead of 30, we could see that leveraging the uncertainty give like better performance and increase the rate of discovery of drug combinations. So that was an interesting observation. And another interesting observation was that basically, even though there were huge batch effects between studies, as we saw, knowledge could still be transferred between studies. And we actually saw that if we were pre-training on one study, performance was increased when performing a sequential model optimization on the other study. And still here, we were careful about the way we splitted the data sets to make sure that the combinations that happened in both studies were not used twice and things like this. All right, so now I will say a word about the prospective application of Recover, and the part where we actually performed in vitro experiments. So in real life, I would say, in the wet lab. And the first step was to actually design the experiments based on the insights from the Silico Evaluation. And so the main takeaways from the Silico Evaluation were that we cannot generalize right away to new drugs or to new experimental settings. And so it's difficult we cannot transfer knowledge right away between databases, but still in the context of SMO, it's possible to leverage existing databases. And here on the left-hand side, you can see the way we set up our experiments. So the gray part corresponds to the almanac study. And so you can see that this matrix is quite full already, so there's not much to search. So we needed to include some new drugs. So drugs 95 to 150 here. And so our search space is the white square basically here. And we were careful about the number of drugs involved because we didn't want to make this space too large because that would be too difficult to search and it would take too many rounds given that we had a limited budget and a limited number of rounds that we could perform. And we didn't want to make the space too small because that would make the problem too easy. So that's for our search space. And you can see that we basically tried to leverage Almanac because there are some drugs that half of the drugs we consider actually come from Almanac. And so we performed 3 rounds of in vitro experiments. So a first round of calibration to understand the differences between the different models and a bit of random exploration. Then the second round was already model guided, but we added big constraints to make sure that each drug was observed in at least one combination. So we basically enforced a lot of exploration. And finally, the third round was basically unconstrained sequential model optimization search, so model-guided search. And you can see that these distributions develop a heavier tail towards the positive synergies. And so basically, we discovered more and more highly synergistic combinations. And this is actually statistically significant when we compare the third round to the random the distribution within the Almanac dataset actually. And interestingly, in the third round, we rediscovered a combination that was actually under study within clinical trials. So it's pazopanib and alisertib, and we also discovered another combination, which apparently is not studied in the literature, but it has the same efficacy as the first combination, but at a lower dose. So maybe it's worth investigating. And we performed a 4th round of experiments, which is not in the archive paper, but we just got the results last week. And as you can see in the last round, this is quite a big jump. So we are doing much better than the third round, which was already statistically significant. And basically, so in this 4th round, out of 30 combinations that were queried, 4 had a synergy higher than 55, 55 being the best synergy that we had discovered so far. And we even discovered one combination with synergy higher than 93. And so we are in the process of validating these findings and doing one last and final round of experiments. And so that's it for the in vitro experiments. And basically, besides the specific applications to drug combinations, I really think the main takeaway from this project is that we really used the in silico evaluation of the models and careful analysis of the out of distribution generalization abilities of the models to inform the design of in vitro experiments and ensure like a smooth and successful interaction between the machine learning and the wet lab. All right, thanks so much. And I'd like to mention that this research is funded by the Bill and Melinda Gates Foundation. And yes, thanks for listening to me.
Thank you, Paul, for the great talk. You can see all the upload. And if there, I think it's time for question. There was one by Dennis that maybe you can start with.
Hello, great talk. I was curious about the noisy data that you're utilizing. Would it make sense to use L1 regularization as opposed to L2, considering there's a large number of noisy data points? Do you think that could be a simple solution to improving the model?
So I'm not sure. Let me go back to the noisy part. So I guess you may want to use L1 regularization in some context where you expect maybe like a few features or like a sparse set of features to be informative about your predictions. In our case, I don't think this is the case. So we of course try to regularize our models with L1 and L2 regularization, but it did not have like a big impact. So here, I think a big part of the noise is what we call aleatoric. That's something that you cannot predict in advance. So I think that's something that we could not tackle with a new regularization or a fancier model. It's just the problem that there is variability from one batch of experiments to the other, and it's due to unobserved factors such as the state of the cells or maybe genetic mutations that we don't know about. Yeah, does that answer your question? Or
yeah. Do you think it would make sense then to maybe do some anomaly detection to remove like the most noisy data points? Do you think that that could be useful?
Yes, definitely. It's actually something that we did, so I did not detail it. But basically, so the bioinformatics team went through the dataset and filtered some examples that were considered bad based on how basically those response metrics looked. So in some cases, the landscape of viability looked quite smooth, meaning that the experiments at various concentrations were somehow consistent with each other. And in some cases, those response curves were very spiky and the measurements were a bit all over the place. So we decided to filter out these specific examples. We did not explore automatic outlier detection, however. But that could be something interesting. And of course, we should be careful because here we want to detect outliers in a way because we want to detect the top 1 or 0.1% of combinations that have extremely high synergy. So we should be careful, of course, to not filter out these examples. But that's an interesting question. Thanks.
Thank you.
I have also a quick question about training. So how did you select the negatives for combination? Did you select them from the combination that were observed to not be synergistic, or just pick at random pair of drugs for negatives?
We actually trained on the whole data set. So the task that we consider is just a regression task. And so previous papers making predictions for the synergy of drug combinations tried various strategies, including, for example, some kind of binarization of the synergy, considering that a combination was synergistic if the synergy was above a given threshold and not synergistic otherwise. In our case, it was not really possible because we wanted to query the really top K%. And if you binarize your input, you cannot rank all the combinations anymore. We did try some oversampling strategies. So because as you saw, basically the really highly synergistic represent only like a tiny part of the data set. So we wanted to oversample them. In practice, it did not change the performance much. So we just did not use oversampling in the end. But that's a good remark. We were concerned actually that the positive cases were underrepresented. So we did try oversampling.
Okay, thank you.
Are there any questions? Hati or Sylvia?
Yeah, sorry I missed the great talk. So I was curious, when you did your drug test splits, did you control for having equal representation of predicted targets or like scaffolds of the chemical structure of the drug? Like, I guess I was curious whether your split data set was truly random, or did it account for some properties of the drug that could be maintained in terms of class balance?
No, we didn't account for properties of the drugs. So we just took all the drugs that were available in Almanac, basically, without differentiating between them. We did explore.
Sorry, go ahead.
No, I just wanted to mention, so we did explore basically the targets of the drugs and try to understand. I have a slide about this in the appendix somewhere. Try to understand a bit more what is learned by the models by looking at the embeddings of the drugs and relating this to known drug targets scrapped from Campbell. And I think this needs further investigation, but we did see that drugs that seem to have similar targets, according to Campbell, seem to cluster together in our embeddings. But that's the only thing that we did related to targets.
I see. Great, thank you.
Yes, so yeah.
Yeah, hi, thanks for the talk. So could you please go back to the slide where you showed the SMO algorithm?
The ensemble?
Yeah, no, the SMO.
All right, at which, in Silico or in vitro?
I think it's a couple of slides back. I think just the previous slide.
Oh, you mean the description?
Yeah, this one. So,
okay.
And yeah, I was just trying to compare this with a general Bayesian optimization framework. So what kind of acquisition function did you use over here?
So the acquisition functions that we used, so was mostly UCB and greedy for the model-based search, and we also compared with the random acquisition function. And I guess so it's really related to Bayesian optimization. However, for example, if you use pure exploration, you would not really consider this Bayesian optimization anymore. And on top of that, here there's nothing really Bayesian about what we are doing, because we will use like we cannot compute the exact posteriori probabilities or things like this. So that's why we try to come up with like a more general terminology. And also the other problem is that if you talk about Bayesian optimization, people will mostly think about hyperparameter tuning, which is not what we are doing here. So those were the considerations that we had in mind.